Hey folks, welcome to our last lesson on diffusion osmosis. So we last time out looked at the effect of osmosis on animal cells when they are put into solutions of different concentration, and we're going to do similar for plants today. And these words are very important to biologists because they describe what the plant cells are going to look like. So we are page 124 in my book, so you know you're going to be a page or two after. The title of the page is What Happens When Plant Cells Are Placed in Solutions of Different Concentrations. And just like the animal cells, you have to be able to predict will water move in or out of the cell based on the words more concentrated. So the big difference between plant cells and animal cells is that plant cells have a cell wall as well as a cell membrane. So your cell wall is fully permeable, it doesn't care what comes in or out, whereas a cell membrane will decide what substances it allows in. So just to remind ourselves, if we were drawing a plant cell, we draw our cell wall, we draw our cell membrane, we draw our nucleus, and we would draw a vacuole. Now there'd be chloroplasts in here as well, but I'm going to omit them for the sake of today. Now my second years currently, when I draw plant cells, say, oh miss, here's your chair and you have a swimming pool, which is the vacuole. So if that helps you to remember what's in a cell, plant cell, please do. So this is your vacuole and this is your nucleus and this is your cytoplasm. So the fact that you have a cell wall means that plant cells can't burst. So they won't burst because of the presence of the cell wall. Whereas animal cells, we saw the last day, will burst. So pause now if you need to. So we have the first scenario. If I put a plant cell surrounded by a solution that is the same concentration as the cytoplasm, so what is that going to mean? So cell water will move in and out at the exact same rate. So if this is our cell, you're going to have water moving into your cell, but also you're going to have water moving out at the same rate because there's no difference in concentration. So the cell will appear to remain intact and it'll look exactly the same under a microscope. And biologists use this word to describe a cell that is unchanging because it's in the same concentration of cytoplasm. So they call it a flaccid cell. So for all intents and purposes, the cell looks exactly the same. So they are flaccid cells. So now the next scenario is if we put a plant cell into a solution that is less concentrated. So you have to remember now, what does less concentrated mean in the context of water? So less concentrated means that there's more water outside of the cell. So if this is our cell, like so, there is now more water outside. So more water here, less water here, and we know from the definition of osmosis that osmosis is the movement of water from an area of high concentration to an area of low concentration. So what will happen is water will now move in to the cell. And the cytoplasm now gets filled up, so it pushes against the cell wall. It's really, I call it a chunky monkey cell. So this can happen when root cells absorb water from the soil. And the word for this is really important. It's called a turgid cell, or that the cell is under turgor. And the SEC like to ask little extra questions on the basis of this word. So in my head, turgid cells, I call them chunky monkey cells. They're really big, fat, full of water looking cells. So a little bit more about turgor. So you have extra little pieces of information. So when the water is moving into the cell, your vacuole becomes bigger. Your cytoplasm swells, it's literally full of liquid, and the membrane is pushed out towards your cell wall. And your cells when they're fully swollen, they're described, as we've said, as turgid. So this all happens because water moves into the cell. So that's the underlying thing that is bringing about turgidity. Now, when we talked about our practical, when we had our visking tubing and we had filled it with glucose or sucrose, and we just had water out here, the water moves in, and one of the measurements that you had was on the basis of appearance, and we had said that if it gets fatter, then it's an increase in turgidity. So here now is the word again in the context of plant cells. So turgid pressure is really, really important for certain types of plants. So I have a love-hate with basil because I'm working from home. That love-hate it's not as bad, but I could have my basil in the morning, water it, and my basil is beautiful and upright, and then by the time I come home later in the day, my basil has fallen over because it has lost all of its water. It's probably too hot where it is. So turgid pressure is what's keeping this upright here. So the fact that the cells are full of water and very plump, it keeps them up. When they wilt, there's not enough water there. This is really important for plants that don't have wood. So trees don't have this issue, but any type of herbaceous plant, a green soft plant, will wilt if it doesn't have enough water. And the last scenario is what happens when we put plant cells into a solution that is more concentrated. So this goes back to 2 lessons ago when I was banging on what the hell does more concentrated mean. So you always have to remind yourself that more concentrated means that there's less water. So if we're putting our cell into a solution where there is less water out here, it means there must be more water inside the cell. So now water is going to leave because of osmosis, and whereas in the animal cell it shriveled and died, this isn't what happens now. So the cell wall will stay intact, that's not going to shrivel, but the cytoplasm itself will shrivel. And you can actually see this if you have red onion cells and you put them in salty water and you look under a microscope, you can actually see them. So our cell afterwards, just looking at the cell, what would it look like? So you'd still have your cell wall, and your cell membrane would actually be just stuck to little parts of your cell wall, but it would be totally shriveled up and you'd have a really small vacuole in there. So this is the example of your cytoplasm shriveling after you leave it in there a while. And again, plant scientists have a word to describe this scenario. They call the cell that it's a plasmalized cell, or that the cell has undergone plasmolysis. So make a note of those, and this is just another version of the diagram. So here's the water leaving, and this is our cytoplasm that it's shriveled. So the cell wall won't shrivel, but the cytoplasm will. And in terms of the little bit of detail, so important thing here, and this is all being brought about because water is leaving the cell. So the cell wall stays intact, your cytoplasm shrivels, and your cytoplasm shrinks and your cell membrane shrivels away from the cell wall. Okay, you should never really use the word it, you should always explain what it is. So here's just the example, water is leaving and the cell membrane, or the cell membrane has shriveled away from the cell. There's not that much water in there. So this is what red onion cells look like if you put them into salt water. You can see that this is the cytoplasm and it's now all shriveled up. So one of the last things I want to look at before we go a little bit further is this was a question that came out about 3 or 4 years ago, and it was based on osmosis. So in an experiment, a student cut some potatoes into small, evenly shaped pieces. She divided them into groups of 10, weighed each group and played them into sucrose solutions with different concentrations. 2 hours later, she removed them, dried them and reweighed them and plotted her graph. So you have to work out if there's a change in mass here, is water going into your cell or out of your potatoes? So because the change in mass is positive, water must be going in. Whereas down here, there is a loss in mass, so water must be moving out. And here is where the concentration of the potato cells and the water is the same. So this is going into, so this is the flaccid scenario, this is the plasmolysed scenario, and this is the turgor scenario. And the questions then they asked, from the graph determine the concentration of the contents of the potato cell. So that's when there's no net movement of water, so C, which is 0.4% sucrose. And then explain in detail why the pieces in solution D to F lost mass. So if water is moving out of the cell, it must be in a more concentrated solution, because more concentrated means less water. So it's in a more concentrated solution, so water leaves the potato cells. Now this question wasn't liked by a lot of candidates, which meant that the SEC front-loaded the marks. So for 2 correct answers, you got something like 17 marks out of 20. So if you see a question that you don't really like, be thankful, because they are going to have to ask you or give you loads and loads of marks. So don't ever be afraid of questions. Always attempt them in some way. You're going to get some marks from it. Now I'm just going to fly away a little bit here, because I just want to look at the last little bit of this. So this is a question that can be asked. Explain the biological basis for the use of high sugar or high salt in the preservation of food. So what that means is, if you have tinned peaches, or if you have ever seen Spam, they can be years and years old and they never go off because they're in sealed containers. And it's really difficult to have them contaminated with bacteria. So it's usually, so if we just see, before we get into it, and there's a space on by the bottom of page 125 to draw this. So if I have a sugary or salty food, so we'll go for salty food here, and I get my little bacteria contamination. Now obviously bacteria don't have eyes and smiley faces, but we're going to label it up anyway. So if there's salty food, it's more concentrated, which means there must be less water here. It means there must be high water in here. And what happens is the water leaves the bacteria and the bacteria will shrivel and will die. Okay, and it's osmosis is how this works. So for you to be able to explain it, the bacteria and fungi are the most common causes of food spoilage, but they don't really work in sugary or salty foods. So both of these cells are enclosed by a cell membrane. The food is placed in your sugary or salty solution, which is more concentrated than the cytoplasm of the bacteria. So more concentrated, less water. And water will leave your bacteria and move into the high sugar or the high salt. Our poor bacterial cells will shrivel and die. And this is the way that food will not be contaminated with loads of microorganisms. So this is your explanation. If you were asked to explain the basis for preservation of sugary and salty foods. So we're going to leave that there. The homework is going to be on the class notebook page. Don't forget to put your RAG notes into your class notes section, and I will be online for Zoom if you need me during this lesson.